we got up to here. And I hope you appreciated the different roles of these soluble mediators of innate antibacterial immunity. Lysozyme breaks down the cell wall, complement will disrupt the outer membrane and also attach complement components. C3b, which is an opsonin, which will favor phagocytosis and liberates these chemotactic proteins, chemotactic fragments C3a and C5a, and antimicrobial peptides, which again will disrupt the outer membrane of gram-negatives. Okay, so the soluble arm of our innate defenses work together with the cellular arm, and what is the most effective for eliminating bacteria are phagocytes, neutrophils, and macrophages. So it's written in quite small letters here, but okay, the different steps are first, attachment of the bacteria to the phagocyte and uptake into a lysosome. So in order for this to happen, of course, the phagocyte has to express receptors on its surface that can recognize the bacterium. So some of these are innate receptors, or for example, okay, the mannose receptor here and the LPS receptor, CD14, can recognize characteristic structures on the surface of the bacterium and favor internalization. Often, these structures on the surface of the bacterium may be hidden or not recognized very effectively by these innate receptors. So, Fc gamma receptors or complement receptors on the surface of the phagocyte are often very important to mediate efficient uptake. And that's why deposition of C3b of complement on the surface of bacteria is often very, very important to favor phagocytosis. So, bacteria can be taken up into phagocytes via these phagocytic receptors. They are internalized firstly into a phagosome, which will then mature into phagolysosome. So maturation is a very important process which has different components which are play different roles in killing bacterial cells. Now, one of them, which is highlighted here, is the secretion of reactive oxygen intermediates into the phagolysosome. So, following binding and internalization of the bacterium, the phagocyte will become activated, and it's going to really upregulate its oxidative metabolism. This is called the oxidative burst in phagocytes after phagocytosis. That's important because it allows all these reactive oxygen intermediates to be produced. And you need to have a lot of NADPH to do this. Okay, so glycolytic metabolism ramps up. The Krebs cycle ramps up in phagocytes after phagocytosis. It gives you a lot of NADPH, and this supplies the electrons to produce all of these different reactive oxygen intermediates through the actions of these enzymes, NADPH oxidase, superoxide dismutase, myeloperoxidase. So you get hydrogen peroxide, hypochlorous acid, singlet oxygen, and hydroxyl radicals. These are all highly reactive and highly toxic. They'll react chemically with DNA and damage it. So bacteria are killed inside phagocytes by the production of this biological bleach. Okay, so you have one oxidative burst. gives you reactive oxygen intermediates. 2, acidification of the phagolysosome. So a type 5 proton ATPase is in the membrane of the phagolysosome, and it will secrete protons into the phagolysosome, so it becomes acidic. This will activate different proteases and nucleases that are also secreted into the phagolysosome. So in order for these to work and degrade the bacterial cell, they need to be activated by low pH. Okay, you also have lipases, DNAs and RNAs. You also get lysozymes secreted into the phagolysosome and antibacterial peptides. Okay, so that's the process in the bacteria, in the killing of phagocytose cells. So this is one of the mechanisms by which some pathogenic bacteria can persist, is they can prevent this maturation process. So if this is prevented, you don't have any of the oxidative burst, you don't have phagolysosome acidification, you don't have the production of all these toxic enzymes that will just pull apart the bacterial cell. Okay, so that's the role of the cellular arm of innate immunity. Adaptive immunity, how do we become resistant to subsequent bacterial infections? In most cases, antibodies are the most important effectors. And they can have multiple effects. So antibodies against bacterial adhesins can prevent the first step in colonization, prevent the bacterial cell from adhering to the epithelium. So this will be important for IgA, isotype antibodies, because these are the ones that are secreted into the mucosa. Okay. Other types of antibodies can prevent bacteria from growing inside the host. So if they bind to the external membrane proteins that are involved in iron transport, for example, then the bacteria can't get any iron and they will not be able to grow. Some antibodies can bind to and inactivate exotoxins. So the bacteria may be able to colonize you, they may be able to grow, but they can't kill you because the exotoxins are inactivated. And many antibodies specific for bacteria are effective because they will coat the outside of the bacterial cell. For example, those that are specific for capsule antigens, they can coat the outside of the bacterium, and that will help complement fixation and phagocytosis. So they allow the immune system, the innate effector mechanisms to function. And only in certain cases, if bacteria grow intracellularly, then cytotoxic T lymphocytes, CTL, can also be important. So bacteria that grow intracellularly, Mycobacterium tuberculosis, Listeria. Antibodies are not so effective or are not so important in immunity. It's mostly T-cell mediated immunity, which allows the infected host cells to be killed. Okay, I'm going to skip this because we just presented it. So now what I'm going to do for the next hour or so is present these 3 examples of bacterial pathogens. 1 gram-positive and 2 gram-negatives. And I'm afraid as we go through, it's going to become more and more complicated because with Streptococcus pneumoniae, you've got 1 bacterial species and 1 type of infection. For Salmonella, we have one species and two different types of infection. And for Escherichia coli, one species, but a bunch of different pathologies depending on what particular virulence factors are present. Okay, so first off, Streptococcus pneumoniae. Okay, as its name indicates, it's responsible for bacterial pneumonia. Can often give otitis in children, and can be very, very serious because it can cause meningitis. So this is a bacterium that can cause invasive infections. Now that means that the bacterium can be found in a compartment of the body that is normally sterile. Okay, so of course, bacterial meningitis, that's an invasive infection. You don't normally have bacteria in your brain. Pneumonia is an invasive infection. It's normally a sterile compartment. Infection of the middle ear without complications, that's not really an invasive infection because it's kind of a compartment that's open to the outside environment, not sterile. Okay, so invasive infections means, meningitis and pneumonia. So in France, 5,000 to 7,000 a year. So who's at risk for these invasive infections? Well, very young children, less than 1 year of age, babies, and then so they're susceptible. And then as you get young children, their immune systems are people that have had contact with this bacterium, they'll develop antibodies and they'll be resistant. That's pretty much how it stays until you get older. So people over 65, as you get older, your immune system kind of wanes, just like everything else, I guess. And older people get more and more susceptible to these invasive infections. It can be a severe cause of morbidity and mortality in older people. Now, so that's the problem. How do you identify this bacterium? If somebody's got pneumonia or meningitis, then they can be identified by streaking the bacteria out on blood agar. And what you're looking for are gram-positive cocci that are catalase negative, okay, so Streptococcus, that show alpha hemolysis on a blood agar plate. So alpha hemolysis is like this. You can see that the blood agar, so these are with, intact erythrocytes that have been incorporated into the agar. So that's what gives the red color, and you have discoloration just underneath the colonies. So in fact, the erythrocytes here aren't really lysed, but the hemoglobin is chemically modified because the bacteria secrete hydrogen peroxide. And so this causes discoloration of the hemoglobin. Beta hemolysis, you would get a very large region discolored at a distance from the bacteria. And in that case, the erythrocytes are really lysed. And so, for example, Streptococcus pyogenes is beta hemolytic. So that's part of the identification. Okay, you have the distinction between pyogenes and pneumoniae because of the hemolysis. So this will give you an indication that you have a Streptococcus pneumoniae, and then this will be confirmed with an API gallery, this time specific for Streptococcus. Now, colony morphology can be variable. They can be transparent or opaque. And as you may know from genetics, you can get rough or smooth colonies of Streptococcus pneumoniae. So the rough colonies do not have a capsule antigen, and they are less virulent. Smooth colonies have the capsule antigen. So what does this remind you of? What should you all think about right now? Rough, smooth, Streptococcus pneumoniae, molecular biology. Very, very old historical experiment. Griffiths and Avery. Does this ring any bells here? No? Nobody at all? Okay, so the Griffiths and Avery experiment is, okay, you've got these smooth bacteria. They are pathogenic. You inject them into mice, they'll be killed. The rough colonies, they don't have a capsule. You inject them into mice, they will live. If you have a mix of live bacteria that are rough and killed bacteria that are smooth, the mice still die because you have transfer of genetic information from the dead cells, dead bacteria, to the live ones. And it was proven that this was via transfer of DNA. This is one of the very first indications that DNA is a genetic material and not proteins. And I am sure you have done this in molecular biology. Yeah. Okay, so that's giving us an indication. These experiments, okay, rough variants don't have a capsule. They are not very pathogenic. It's giving us an indication the capsule is very important for pathology. And when you look, what is rather unfortunate is there are more than 90 different serotypes that can be defined on the basis of variants in the capsule antigen. So you think, how is it possible? How can we use this to produce a vaccine, because we can't put 90 different antigens into the vaccine? It's just too much. Well, fortunately, the invasive bacteria are not evenly distributed over these 90 different serotypes. So if you take the capsule antigen from the 13 serotypes that are most prevalent in invasive infections, then you get a vaccine. So these 13 serotypes are responsible for more than 95% of invasive infections, and this is what's used in the Prevnar 13 vaccine. Okay. So that's the pathology. That's how we can identify the bacterium and control or prevent infection. What are the different steps in the pathogenic process? Adhesion invasion. This is mediated by pneumococcal surface protein C, PSPC, and it works in a very clever way. Okay, here are some epithelial cells on the basal layer. Right. In the respiratory mucosa, then our gram-positive S. pneumoniae will be here, okay, on the apical layer. And PSPC is going to bind to a host cell protein called the polyimmunoglobulin receptor, PIGR. Now, PIGR is part of our host defense. Its role is to pick up immunoglobulin A, secreted by plasma cells underneath the mucosa. Going to be in a vesicle here with dimeric IgA. And it transports dimeric IgA to the apical membrane. Then this will release IgA. And then the receptor will recycle down to the basal surface. So what Streptococcus pneumoniae does is it binds to the poly-Ig receptor and uses it as a kind of molecular taxi to bring the bacterium across the epithelial layer and release it on the basal side. That's a very specific mechanism for traversing the epithelial cell barrier. So once it gets down here, the bacteria are going to find themselves in the extracellular matrix. And they're going to degrade it by using the hyaluronidase. So they'll secrete this enzyme, going to degrade the extracellular matrix, and that will allow bacteria to grow. So adhesion, invasion, colonization. Next step is the bacteria have to grow effectively in the host. All pathogenic bacteria need iron, so this is the role of PSPA, which will bind to lactoferrin. So it doesn't use a siderophore, it picks up one of the host cells' iron transporting, host organisms' iron transporting molecules. And then got to evade the host immune response. This is the role of the capsule antigen. So these bacteria will all be surrounded by capsule antigen, and that will inhibit phagocytosis and inhibit complement deposition. And finally, in order to really harm the cell or harm the host, it's going to have some kind of toxins. Streptococcus pneumoniae secretes pneumolysin. So this is a small protein. When it's secreted, it's a monomer. And when it inserts into a phospholipid membrane, it oligomerizes and forms a pore. So it lyses the cell membrane. Now you might wonder, okay, why don't the bacteria just lyse themselves with this protein? In fact, its oligomerization is induced by binding to cholesterol. So cholesterol is specific for eukaryotic cells. Bacteria don't have any cholesterol. So this toxin is specific for lysing the host cells. Okay, so that's it. Those are the different virulence factors in Streptococcus pneumoniae. Okay, so how about Salmonella? Now here we go, Salmonella, one of the Enterobacteriaceae. Commensal, okay, which are all commensals. Some of them can be strict pathogens in humans. Salmonella will be a commensal bacterium in birds and in reptiles, but strict pathogen in humans. And some Enterobacteriaceae can be opportunistic pathogens, Proteus E. coli. So something you'll need to know about Salmonella phylogeny is this. So officially there are only 2 species in the genus Salmonella. Salmonella enterica and Salmonella bongori. Now, often you hear people talk about Salmonella typhi, Salmonella paratyphi. these are subtypes of Salmonella enterica. Okay, so whenever, and actually I might say this, but because it's quicker. So all of these different subtypes are part of Salmonella enterica. So how do you identify them? Firstly, by so they cause salmonellosis, gastroenteritis, and can cause typhoid fever if they are transported into the blood. So mostly you'll be using coproculture to identify salmonella, sometimes hemoculture. And then you can identify them on selective media. So this is SS medium. It's going to be selective for gram negatives. In this case, it doesn't have eosin like the EMB plates, but these SS plates have got bile salts, which will inhibit the growth of gram positives. And it's differential for Salmonella and Shigella, which is a name here, SS. Okay, so it's got lactose and it's got neutral red as an indicator. So if bacteria, if you've got lactose positive bacteria, the colonies should be red. It's also got thiosulfate and ferric iron. So if bacteria produce H2S, then you'll get a black identification. So Salmonella are going to be lactose negative, sulfide positive, so the black colonies. And Shigella will be lactose negative, sulfide negative. So that's what you do. Get your coproculture, dilute it out, streak it out on an SS plate. And then to confirm your diagnosis, you'd pick these black colonies and put them on an API gallery, for example. And then this would give you an identification at the species level, Salmonella enterica. After that, you would want to identify the subspecies by serotyping. And serotyping is going to be based on the variability of the bacterial O, H, and possibly K antigens. So everybody remember what these are? O antigen is what? I know it's a long time ago now. Okay, these are surface structures on the surface of the bacterium that can be bound by antibodies, and they are useful for identification. Because the bacterium will only have one type of this on its surface, and it will be different between different strains. Yeah, O antigen, LPS. O is for O's. It's the variable sugar repeats on the external part of the LPS. That's what's being recognized. H, heat sensitive. The flagellum on the bacterium, if present. K is for capsule, if it's present. Okay. Everybody remember how serotyping works with the agglutination test? Here's an example of a result that you might see at a low power microscope. One of these is positive, one of these is negative. Okay. Who says this is positive result? Who says this is the positive result? Maybe one person. Who doesn't remember how this works? Who are not actually people, but are just pod clones grown in a pod that cannot answer yes or no to a question? Most people in the room, right? Okay, so I don't know. Sometimes it's difficult for me to understand because nobody answers to the specific question. Who doesn't know? Who doesn't remember? Some people. It's almost like all the rest of you do remember, but you just want to keep it a secret. Okay, so just for a quick reminder here, you will have bacteria. They might have a particular O antigen, O1. Oop. Okay, so they have a particular sugar repeat in the LPS. And you're going to incubate these bacteria with beads that are coated with antibodies specific for the O1 antigen. So in the presence of these beads, the antibodies will bind to the O antigen on the surface of the bacterial cells, and the whole thing will agglutinate. So the beads and the bacteria all bind together in a big clump. That gives a positive reaction. You see a clump and it all goes together. That's what you see here, okay? It's all agglutinated, the beads and the bacteria together. So if the antibody on the beads does not recognize the antigen on the surface of the bacteria, then the beads just stay in suspension. It's homogeneous, which is what you see here. So this is positive, this is negative in an agglutination test. So you get a suspension of the bacteria, get a suspension of the beads, mix them up, leave it on the bench for 20 or 30 minutes, and then look down the microscope and see whether there are presence of these clumps. So it's rapid, it's easily read as a result for positive or negative, and the only drawback is you need a whole bunch of different beads with different antigens, antibodies for the different serotypes. So the classification of Salmonella subspecies is based on this, which is called the Kaufman-White classification. And it's based on the different O antigens, the different H antigens, and not so much the capsule. VI is for virulence, and it's the capsule antigen. Okay, so O antigens. These will define the group. And the H antigens pretty much define what these the subspecies are. So H antigen phase 1, phase 2. Phase 1 is motile, so these bacteria have flagella. So this is the flagella antigen. And phase 2 is when they are non-motile. This is some other kind of protein on the surface. Don't have to worry about what it is. Phase 1, this is the flagellum. Okay, so you don't have to remember this classification. You just have to know that classification of subspecies of Salmonella is based on serotyping. Okay, and the main ones are the O antigen, the H antigen. The capsule is mostly absent apart from paratyphi C and in typhi. Okay, so that's the bacterium. What are the pathologies? The most frequent one is Salmonella gastroenteritis. So if you ingest Salmonella enterica, then you have diarrhea, stomach cramps, and fever. 12 to 36 hours after the inoculation. So fever is telling you that you have bacteria that have arrived in a compartment that's normally sterile in your body, so it's an invasive infection. So Salmonella enterica enteritidis, you can get this from eggs and derived products like mayonnaise, things that are made out of eggs without cooking. Typhimurium can come from meat products. So, okay, Salmonella enterica enteritidis, it's commensal in chickens, So when eggs are laid, they can be contaminated with this bacterium from the chickens. Typhoid. So as well as that, the other type of pathology you can get from Salmonella is typhoid fever. Now, what this is characterized in this case, you start out with the gastroenteritis, but you also have bacteremia. So you have bacteria in the blood and a systemic infection, not localized. So this can be more serious. It's estimated that there are 21 million cases per year. The case fatality ratio is about 1%, so that means you have about 200,000 deaths per year due to this bacterium. What can you do about it if you've got Salmonella gastroenteritis? Your doctor will give you kind of symptomatic treatment, lie down, drink a lot of water, wait till you feel better. If you have fever, take paracetamol. If you have a typhoid fever, then you'll need to have antibiotics, fluoroquinolones and third-generation cephalosporins. Now, Salmonella gastroenteritis used to be the king in France. So in 1996, more than half of the foodborne disease outbreaks in France were caused by Salmonella. These days, I don't know, rather losing some popularity here. This is the light blue line, only 10%. It seems like Staphylococcus aureus is having having a bit of a breakthrough in Bacillus cereus. Don't particularly know why this tendency has changed, to be honest. Okay, so what is the disease process? It's an invasive infection. People get fever. This is and stomach cramps. You don't get that for, cholera doesn't cause the same types of symptoms, for example. So what is happening? So the bacteria arrive in the lumen of the gut, and they induce their uptake by bacterial proteins coded on Salmonella pathogenicity inode 1. So they induce their uptake. They're transported across the epithelial barrier. Then they're going to be phagocytosed by macrophages, which would normally kill them, right? Normally this is going to be a career-ending move for bacteria to get phagocytosed because they're going to be killed. But proteins on another pathogenicity island, SPI2, allow the bacteria to survive within the phagocyte. So SPI1 does this, causes a non-phagocytic cell to take them up. SPI2, how does that work? SPI2 codes for a type 3 secretion system, which will cross the phagolysosome membrane and allow the secretion of different effector molecules into the cytosol of the phagocyte. And what these are going to do is prevent phagolysosome maturation. So all of these things that are dangerous for bacteria, the oxidative burst, reactive oxygen species, activation of proteases, nucleases, and secretion of antibacterial peptides, these things don't occur because of the action of the SPI2 secretion apparatus and the effector molecules that get injected into the cytosol of the phagocyte. So SPI2 proteins are not generally expressed. Their expression is controlled by this system, PhoP, PhoQ, which is a kind of detection system that the bacterium uses to tell it whether it's inside a phagocyte or not. So PhoP, PhoQ will also activate the expression of SPI3. So this codes for magnesium siderophore, because inside the phagolysosome there's not very much magnesium. So in order to grow, they have to get magnesium. So how does this Pho-P, Pho-Q system work? So Pho-Q is a membrane molecule, membrane protein. And in conditions of low magnesium and low calcium, it's going to be activated. Once it's activated, it phosphorylates Pho-P. And Pho-P is a transcription factor, which will upregulate the expression of the virulence genes, SPI2 and SPI3. So after phagocytosis, Pho-P, Pho-Q tells the bacterium, you've been phagocytosed. This allows the expression of the virulence factors that allow the bacterium to survive inside the phagocyte. So all of that is pretty nasty. But what is the virulence factor that allows the bacteria to cause typhoid fever? Gastroenteritis is unpleasant, but it's very, very rarely a life-threatening condition. Typhoid fever, this is the one that is dangerous. So if you look at the subspecies that cause gastroenteritis, Salmonella enterica enteritidis, and Salmonella enterica typhi that causes typhoid fever, okay, they both have SPI1, SPI2, and Pho-P, Pho-Q, and SPI3. Okay, so both of them traverse the gut epithelium. Both of them can survive inside macrophages. But that's not enough to allow this bacterium to cause typhoid fever. So the big difference between them is this one, okay? It's SPI7. SPI7 codes for the protein that allows the synthesis of the capsule antigen. So once again, as for Streptococcus, its capsule antigen is a major, of Streptococcus pneumoniae, capsule antigen is a major determinant of pathogenicity. Okay, so let's see if you've understood this. Now, read the question carefully. We have 4 propositions, A, B, C, and D. And the question is, which of them are true for Salmonella enterica, but false for Streptococcus pneumoniae? Everybody decided? Yeah? Okay, so A, codes for a toxin that lyses host cells. Who says that's correct? Who says it's wrong? You're right, it's wrong. So it's true for Streptococcus pneumoniae, false for Salmonella. Bacterium can inside in phagocytes. Is that correct? Yeah. Who says it's false? Okay, it's true for Enterica and not true for pneumoniae. The capsule antigen is an important virulence factor. True for Salmonella enterica, false for Streptococcus pneumoniae. Who says that's correct? Who says no, it's not correct? You're right, it's not correct. Important for both of them. D, the bacterium has a specific mechanism that allows it to cross the epithelial barrier. True for Salmonella enterica, but false for... Says that D, correct? D, not correct. You're right, it's not correct. Both of them have a specific mechanism that allows it to be transported across the epithelial cell barrier. So in Streptococcus pneumoniae, it's PSPC, and in Salmonella enterica, it is the proteins encoded on SPI1. Okay, so this leaves us 30 minutes to think about Escherichia coli. So as you all know, mostly Escherichia coli are not harmful. They are commensal bacteria, and it's probably the most frequent or the biggest contributor to the aerobic gut flora. But some strains can be pathogens. Okay, basic characteristics of E. coli: gram-negative coccobacilli, lactose positive, indole positive, urease negative. And if you want to look below the species level, there are a whole bunch of serotypes based on, once again, O antigen, LPS, H antigen, flagellum, and the capsule antigen, K. So this kind of designation here identifies a particular serotype of E. coli. Now, different strains of E. coli can cause non-enteric infections. So you can get urinary tract infections from E. coli. We're not going to talk about them. We're going to concentrate only on enteric infections. Oh, just remember, the choice here depends on the adhesin that is expressed by the bacterium. Okay. Escherichia coli, often people think, you know, it's a commensal, sometimes it can cause infections, but it's not really a big health problem. Well, in fact, it is, because it's the second most frequent cause of diarrhea in children worldwide. And if you don't have access to a lot of clean water, then this can be fatal, because children, especially small, young children, can very quickly die from acute dehydration. So the disease burden due to E. coli is really rather high in the world. Now, depending on the type of the pathovar, you have different types of pathology. So, and that's what we're going to try and understand here. Enterotoxigenic E. coli cause childhood diarrhea and traveler's diarrhea. Oh no, enteropathogenic E. coli cause childhood diarrhea. Enteroinvasive E. coli give you kind of dysentery, and enterohemorrhagic E. coli give you a hemolytic uremic syndrome. So the point is to try and understand how these different E. coli are causing different diseases. What are the mechanisms involved? So we're going to start with enterotoxigenic E. coli. It's what you catch if you go on holiday to, I don't know, Marrakesh or Bombay or something, simply because you'd have had the diarrhea caused by E. coli when you were a kid, and all the strains circulating in Nantes are about the same, so you're immune to them now. If you go somewhere else, there are different serotypes around with different antigens. Your IgA antibodies in your gut don't protect you against the strains that are present in different countries. So you go somewhere else, you get colonized by different bacteria, and then you're susceptible to them. So what's happening? First step, attachment to the epithelial cells in your gut. These are fimbrial adhesins that mediate this attachment. There are many different types, but they're antigenically variable. So if you've had this before, you get antibodies against the particular fimbrial adhesin. You're immune to those types of bacteria, but other types can still colonize you. Then they'll secrete an exotoxin into the epithelial cells. So this is I just have to try and remember this. Okay, so there are two types: heat labile toxin (LT) and heat stable (ST). So the heat labile toxin, it's a protein, is very much like the cholera toxin. Works in the same way. So it's an AB toxin. It's transported into the enterocytes. It will cause secretion of chloride and watery diarrhea. So you have a non-invasive infection, and the symptoms are provoked by an AB type toxin. So, two types of virulence factor, one for adhesion and one coding for the exotoxin. Very simple. And they're on plasmids. Enteropathogenic E.coli. So what's happening here? It causes diarrhea in infants. First step is adhesion. Firstly by a fimbrial adhesin, and then very, very tight attachment by this intimin adhesin. So the bacterium really sticks onto the surface of your enterocytes. Then it's going to, and this attachment here causes reorganization of, no, okay, that's not exactly, it's not strictly speaking the attachment. Then this bacterium is going to inject bacterial proteins into the enterocyte, which cause rearrangement of the actin filaments. So you get something that looks like this. And the overall effect of that is if you have epithelial cells in the gut, they have a brush border. Okay? You look at them by electron microscopy, you have microvilli. If you have enteropathic E. coli that bind on the top of them, all of this actin cytoskeleton here will be reorganized, and the surface of the enterocytes will become flat. And the net result of that is that nutrient uptake is very, very poor. Okay? Because when you have the brush border like this, you have a very, very high surface area, so nutrient absorption is efficient. If your enterocytes have a flat surface, the surface area is much lower, so nutrient absorption is poor. So if your enterocytes are not absorbing glucose efficiently from the starch you've eaten, all the gut bacteria that you have more food to eat. Okay, they've got more sugar to digest, and this will give you kind of unpleasant diarrhea. Okay, so you have a poor nutrient absorption, the bacteria have more to eat, and your digestion will be perturbed. Okay, so once again, non-invasive. The bacteria stay on the luminal side of the epithelium and are not present in a sterile compartment of the body. Enteroinvasive E. coli are rather different. So if you have an enteroinvasive, an infection from enteroinvasive E. coli, you'll have a fever, diarrhea, but this time you'll have blood in your stools. So this is telling you that the intestinal epithelium has been damaged. How does this happen? First step is the bacteria adhere, then they're taken up into the epithelial cells, then they escape. So they persist inside the cytosol, and then they're transferred from cell to cell. So these bacteria don't secrete exotoxins. However, what you do have is LPS, endotoxin here, which is in the presence of components of the cytosol. It's not where it should be. So you'll have tissue damage caused by inflammation. So these cells that are infected will be killed. You have a lot of endotoxin here, so neutrophils will you'll have inflammation and damage in the intestinal epithelium. And the last one that we need to think about is Enterohemorrhagic E. coli, which is the most severe form. So there have been several epidemics in developed countries. So this is not a problem in developing countries. And most cases are in children under 15 years old. And in most cases, the source is from contaminated beef that has been undercooked. Okay, so what's the transmission source here? I mean, I didn't talk about this, but, you know, this one, Enteropathogenic E. coli, traveler's diarrhea. This is from the E. coli that are circulating in the human population in the country you're visiting. And it's getting onto your food by poor hygiene from the person who's prepared it. Okay, in the shop or in the restaurant where you've had something to eat. So, same for this one really. Of course, children can be a bit dirty sometimes. This one is a little bit different. Okay, so most of the time the origin is not a human source. It comes from cattle, because the pathogenic E. coli are endemic and non-pathogenic in cattle. So when the animals get slaughtered, some of their fecal matter can contaminate the meat. And that can become dangerous if you have something like a hamburger, okay? Because you've got the meat, it's all ground up. Bacteria that are on the outside can be transported into the middle of the hamburger. And when it's fried up, it's only cooked on the outside. So bacteria that are in the middle can be protected from the heat. So hamburgers can be dangerous for this one. Like, I don't know, steak tartare would probably be the worst thing to eat if there's a risk of enterotoxigenic E. coli. A steak, like a bavette just cooked quickly on each side, probably not very dangerous, because the contamination will be on the outside, and just cooking on the outside will be enough. Okay, but it's kind of like these ground meat products are the ones that could present a risk. So if you get that, you'll have bloody diarrhea, and in some cases, hemolytic uremic syndrome. Maybe up to 10% of people who get infected. That's hemolytic anemia and renal failure. So it could be 10% of cases. And this is probably the most common cause of acute renal failure in young children. Can cause neurological complications, and you can possibly have long-lasting sequelae in about 50% of people who survive. It can be fatal because if your kidneys are destroyed, then you will die unless you get a transplant. And one of the things that is worrying about this is that antibiotics do not really show clinical benefit. It's very, very difficult to treat. And one of the things that is done with children who have hemolytic uremic syndrome is to, what's the word I'm looking for? Plasmapheresis. So why is this happening? What happens here? Well, the first thing is that you'll have attachment onto the surface of enterocytes, then very, very close binding through, once again, this intermin adhesion expressed on bacteria, or by the bacteria. And then, so the first part is like Enteropathogenic E.coli, right? Yes. And as well as that, what they will do is they will secrete an exotoxin into the enterocytes. So this is called Shiga toxin. And this is going to be secreted into the enterocytes, will kill them. Some of the Shiga toxin will also find itself in the blood. And it is very, very toxic to kidney cells. So this is a case where one of Koch's postulates is not respected because bacteria can only be found in the gut. It's non-invasive, but the pathology occurs in the kidney, distant site, because of the toxin which passes through the blood. So that's why plasmapheresis is used as a therapy. The toxin is in the blood of the child who's got the hemolytic uremic syndrome. You take out all the plasma of this person, of this patient, you replace it with healthy plasma. So you take the toxin out and you replace the plasma with something else. So why is it that this is only a problem in developed countries? It's because, okay, in developing countries, all the kids get infected with Enteropathogenic E. coli, causing diarrhea. They get antibodies against the intermin. So that means that when they're adults, they'll be resistant to these bacteria. Okay? In developed countries, we're susceptible because we have not had infantile diarrhea caused by Enteropathogenic E. coli. So because the adhesion process is the same for Enterohemorrhagic E. coli, we're susceptible. And the particular thing here is that the Shiga toxins that are responsible for the pathology are coded on a prophage genome in the bacterial chromosome. And that's the reason why antibiotics are not effective, because fluoroquinolones cause genomic stress in bacteria, and this will activate the expression of the toxin from the prophage genome. So if you treat somebody with hemolytic uremic syndrome with antibiotics, this is going to increase the expression of the toxin. So you can kill the bacteria, the pathogenic bacteria, but before they die, they can secrete a lot of toxin. So that's why there's no clinical benefit in antibiotic therapy in these infections. Okay, so 10 minutes to go. So the latest big outbreak of this bacterium in Europe was an outbreak in Germany in 2011. There were 4,000 cases, 50 deaths. This is despite, very high quality medical care. So more case fatality ratio higher than 1%. Unusually, there was no link to beef amongst the people who got infected. And it was caused by a new serotype, O104H4. So it seems like, in this case, the toxin genes have newly arrived in this strain of E. coli and caused the appearance of a new strain of enterohemorrhagic E. coli. So that's it for today, I guess. Any questions about pathogenic bacteria since we have time? Ah, tiens. Wait. I missed this one out, I guess. Okay, so. Okay, no, sorry, that's not it. Yeah, I thought just two more things. Yeah, I was kind of surprised. So, enterohemorrhagic E. coli, how is it diagnosed? Okay, you present at the hospital, you've got bloody diarrhea, you have a fever, you'll get a copper culture. Firstly, look for copper culture on medium like EMB or something like that, looking for Escherichia coli. And then you're looking for lactose positive colonies. Confirm the identity with an API gallery, and then use serotyping to get the O and the K genotypes, K serotypes. Treatment, okay, if you have enterotoxigenic or enteropathogenic E. coli, oral rehydration. In severe cases, intravenous, if you've got a very small child with a risk of acute dehydration, they'll get a perfusion. Invasive infections, enteroinvasive E. coli and typhoid fever, then you get antibiotics. Fluoroquinolones as third-generation cephalosporins, but not enterohemorrhagic E. coli. No clinical benefit. Okay, that was really the end. So that's it. No microbe of the week tonight because I have a grand deadline on the 1st of December. We'll have one more for virology, and then the last lecture will be the final results.